So we're very excited today to have with us Xiaoyan Yi and Priscila Zimath, both PhDs, both postdoctoral fellows at the ULB Center for Diabetes Research. And Xiaoyan, she's a postdoctoral researcher, as I mentioned, at ULB in Brussels, Belgium, working under the mentorship of Professor Desio Zirik. She obtained her PhD in biomedical science at the same center under the supervision of Professor Miriam Knopp and also Dr. Zirik, beginning her doctoral studies in September 2019. With a background in bioinformatics and molecular biology, she's developed expertise in large-scale genomics data analysis, and her PhD research focused on integrative omics of human islets to investigate the pathogenesis of diabetes, particularly glucolipotoxicity in type 2 diabetes, and her current work explores shared mechanisms underlying autoimmune diseases at the level of the target tissues. And Priscila Zimath, PhD, she's also a postdoctoral researcher, again at ULB, conducted her PhD in the Department of Pharmacology at the Federal University of Santa Catarina in Brazil, where she investigated the effects of glucocorticoid mometasone furoate on glucose and lipid homeostasis of rodents. Following her doctoral training, she joined the Center for Innovation in Preclinical Studies in Brazil, focusing on preclinical research in drug efficacy, safety, and toxicology. And then in 2023, she moved to Belgium to pursue her postdoctoral training in type 1 diabetes research. And her current research explores the complex crosstalk between pancreatic beta cells and the immune system in the context of type 1 diabetes. Thank you again for joining us. This is our second recent talk from the Zirik Lab, which is well known as a powerhouse of type 1 diabetes research in Europe and beyond. Thanks so much for joining us.
Thank you for your introduction, and thank you again for joining us. We're excited to share our recent work published 2 months ago in the Journal of Autoimmunity. Many thanks to all these co-authors and the Sugar Science for the opportunity to present and discuss our work here. I will start with a dialogue between beta cells and the immune system in type 1 diabetes. Beta cells sense danger signals through pattern recognition receptors and cytokine receptors, and stressed beta cells respond actively by upregulating MHC class 1, which makes beta cells more visible to the immune cells and secreting chemokines and cytokines that recruit immune cells to the beta cells. Once immune cells, such as macrophage, dendritic cells, and T cells arrive, they further amplify inflammation by releasing interferons, interleukins, and TNF. So this established a feed-forward inflammatory loop in which beta cells become active participants and even amplifiers of their own destruction, rather than passive targets of autoimmunity. And to investigate the underlying molecular mechanisms, we analyzed beta cell transcriptomes using publicly available single-cell RNA-seq data from the Human Pancreas Analysis Program. These included samples from the non-diabetic donors, single autoantibody positive donor, and multiple autoantibody positive donors, and the established type 1 diabetes. Interestingly, when we compare autoantibody positive donor with the non-diabetic donors, we observed interferon signaling and antigen presentation signalings are upregulated only in the donors with multiple autoantibody, as you can see here. And HLA class 1 genes appear to be the most upregulated genes compared to non-diabetic donors. This is not observed in those with single autoantibody. So this upregulation is maintained and even enhanced in beta cells from type 1 diabetes donors, as you can see here, and with the more upregulated HLA class genes here. So these findings suggest interferon signaling emerges only after advanced autoimmunity, potentially acting as an amplifier of immune beta cell dialogue. In this phase, beta cells appear to play an active role rather than being passive targets. In the present study, we focused on the target tissues of 4 autoimmune diseases: type 1 diabetes, Hashimoto's thyroiditis, multiple sclerosis, and rheumatoid arthritis. Our rationale for selecting these diseases include several considerations. Pancreatic beta cells and thyroid follicular cells are specialized hormone-producing cells and may share intrinsic molecular traits. Beta cells and neurons exhibit transcriptional similarity, including shared regulatory patterns involving splicing factors and splice variants. Type 1 diabetes, rheumatoid arthritis, and multiple sclerosis share genetic risk loci and display convergent inflammatory pathways in the target tissue level. In our recent review of the GWAS catalog, which showed that approximately 80% of the risk genes are expressed in the target tissue level of type 1 diabetes and Hashimoto thyroiditis, with a significant overlap in risk genes between these two autoimmune diseases. Finally, therapeutic strategies across autoimmune diseases aim to reduce local inflammation, protect target tissues from apoptosis, and promote regeneration. So analyzing these conditions together enables the identification of conserved disease-driven programs and reviews common patterns of target immune cell crosstalk. To achieve this goal, we compared the global transcriptomic similarities across the target tissues of these autoimmune diseases using the read ribbon pipeline, which is an enhanced implementation of rank-rank hypergeometric overlap analysis. This method allows for the comparison of two differential expression signatures, for instance, disease versus control, to identify the commonly regulated genes in either upregulated or downregulated quadrants. What we can see here, all these target tissues we analyzed shared a set of upregulated genes, as you can see the signal here, with the strongest similarity observed between type 1 diabetes and Hashimoto thyroiditis. So this is biologically relevant, as both are endocrine diseases and involving epithelial hormone-secreting cells. To gain deeper insight into these commonly upregulated genes, we performed functional enrichment. Our results show that the top 20 overlapping enriched pathways include interferon signaling, including both types 1 and 2, cytokine signaling, and immunoregulatory interactions between lymphoid and non-lymphoid cells. So these findings emphasize the central role of interferon signaling in mediating the crosstalk between immune cells and target tissues in autoimmune diseases. We next calculated interferon-stimulated gene signature scores in the target tissues of type 1 diabetes and Hashimoto thyroiditis. The IST gene panel consists of 53 genes specifically induced more than threefold by interferon alpha and interferon gamma, but not by interleukin-1 beta or TNF alpha, as identified from our previous study using NDOC beta H1 cells exposed to individual cytokines. By using publicly available bulk RNA-seq datasets from the target tissues of type 1 diabetes and Hashimoto thyroiditis and their respective control donors, we found that IST scores are elevated in both pancreatic beta cells and thyroid cells compared to their respective control donors, indicating a shared interferon response that may contribute to tissue-specific damage. To further assess the natural progression of type 1 diabetes, we applied the same IST scoring approach to the HPAP single-cell RNA-seq data, comparing non-diabetic donors, single autoantibody positive donors, multiple autoantibody positive donors, and the established type 1 diabetes donors. What we can see here, the IST score began to increase in the donors with multiple autoantibody positive donors and reach the highest levels in beta cells from established type 1 diabetes. So these results reinforces our earlier finding that interferon signaling emerges during late autoimmunity and acts as inflammatory amplifier at the B cell immune cell interaction. We are very curious whether HLA class 1 upregulation, which makes beta cells more visible to the immune cells, is interferon specific or a general stress response? To address this question, we analyzed HLA, B, and C gene expression using publicly available bulk RNA-seq datasets generated by our lab previously and our collaborators, involving datasets of human islets exposed to palmitate, high glucose, their combination, and another ER stressor, brifidin A, which blocks ER to Golgi trafficking, and interferon alpha. Notably, we found that only interferon alpha, you can see here, but not metabolic or ER stress induces upregulation of HLA class 1 genes in human islet cells, indicating that this is not a general stress response, but a pathogenic features of autoimmune activation. And to validate this observation, we, in a second disease relevant target tissue with the colleagues at ULB, we differentiated human embryonic stem cells into thyroid follicular cells. And those cells were then exposed to interferon alpha or interferon gamma for 24 hours. Transcriptomic analysis revealed that interferon alpha and interferon gamma formed distinct clusterings, yet both strongly upregulated interferon signaling, peptide loading, and antigen presentation under both treatment. So these results confirm our conclusion that interferon acts as an amplifier of target tissue inflammation in follicular cells, similar to its role in beta cells. Finally, to identify the potential therapeutic drug candidate, for the commonly upregulated genes, many are related to the interferon alpha or interferon or inflammatory gene signatures. We applied the connectivity map drug repurposing. This resource contains transcriptomic profiles induced by tens of thousands of small compounds, which allowing us to match our disease signature to potential therapeutic targets. So interestingly, FGFR inhibitors have emerged as promising candidates to reduce the shared upregulated disease signatures, as you can see here. Based on literature review, we have selected the FGFR1 inhibitor for further study. So from now, Dr. Zimath will continue the second part of our study, focusing on drug validation in pancreatic beta cells for potential type 1 diabetes therapy. Please, Priscila.
Thank you, Xiaoyan. So to focus a bit more on the drug repurposing, it is a process of identifying new therapeutic uses for previous existing or shelved drugs. So basically, this drug repurposing process takes from 10 to 20 years. Basically the regular drug discovery process takes from 10 to 20 years and involve several steps such as target identification and compound screening, deep in vitro and in vivo studies, and as well as a deep toxicity and safety checking, first in rodents, then in primates, and ultimately in humans. While the drug repurposing process has as advantage the reduced development time, so between 3 to 12 years, the lower development cost, as well as the improved success rate. And also the safety is already checked before. So as Xiaoyan mentioned, by using bioinformatic analysis, we are able to use as a filter these disease-specific molecular signatures to be able to identify and discover a new therapeutic use from previous drugs to now protect beta cells. Next. Thank you. And this drug repurposing process is very important in type 1 diabetes. So in this image that was obtained from the type 1 diabetes trial net study group, show all the drugs that are being tested or are already in use to prevent the progression of the disease. And as we can see, most of the drugs that were investigated for the treatment of type 1 diabetes were first used in other autoimmune conditions, mainly rheumatoid arthritis. For example, the drugs anakinra, etanercept, and most recently the JAK inhibitors. So focusing in the JAK inhibitors as an example, baricitinib, as I mentioned, was first developed to be used to treat rheumatoid arthritis, and most recently was also used in 2019 to treat severe coronavirus disease. And given the preclinical data from analysis of non-obese diabetic mice, and given also this efficacy of the JAK inhibitors in other autoimmune diseases, the baricitinib was investigated in patients with new onset type 1 diabetes, and it shows to preserve beta cell function and improve the metabolic measures. So these inhibitors of JAK1 and 2 isoforms impair cytokine-induced HLA class 1 expression in islets, impair CD8 T cell activation, and also block the communication between the beta cells and the CD8 T cells, preventing the beta cell death. Next. So, based on the bioinformatic analysis, in this paper, we focus in the fibroblast growth factor receptor inhibitors. So, the FGF family comprises 23 members, being 18 ligands that act directly to its receptor, that are the FGFR1 to 4. So these receptors are tyrosine kinase that after being activated and phosphorylated, they activate downstream pathways such as STATs, AKT, and MAP kinases. And in physiological process, this FGF family is very important in a process such as proliferation, survival, migration, differentiation, and metabolism. And the FGF family is expressed in many cell types. So in the clinic, the FGF inhibitors, they are used to treat some diseases that are driven by the dysregulated FGFR signaling. So basically, some types of cancer, as the bladder, colorectal, and lung cancers. But most recently, the FGFR1 pathway was shown as being overactivated in autoimmune diseases such as rheumatoid arthritis, multiple sclerosis, and lupus. And some studies show that the FGFR1 inhibitor has a potential therapeutic effect in vitro and also in animal models that mimics these autoimmune diseases. But the FGFR1 inhibitors were never been tested before in type 1 diabetes and beta cells. So then, basically, based in this information, we check our own data sets and also HPAP data sets, and we observe that human beta cells express only the FGFR1 isoform at significant levels. So among the FGF receptors, we select the isoform 1 to continue our study. Next. And to answer what is the role of FGFR1 inhibitor in type 1 diabetes, we use endoC-beta-H1 cells that are insulin secreting cell line. And we treat these cells with the FGF1 inhibitor PD173074. That is a ATP competitive inhibitor of FGFR1. And we test 3 different concentrations. And after 2 hours, we expose these cells to interferon alpha or interferon gamma, as interferons are the main drivers of beta cell damage in type 1 diabetes. And after 24 hours, we perform some analysis. We also validate our data, the main findings in stem cell-derived islets and human islets. Next. So we can see in graphs A, B, C, and D that interferon alpha and interferon gamma induce the expression of pro-inflammatory genes HLA-ABC and CXL10, but also induce the expression of the protective genes PDL1 and HLA-E in figures A to H. Next. The green bars show the dose response data from with the FGF inhibitors, and we could see that the FGF inhibitor decrease interferon-induced pro-inflammatory genes in a dose-dependent manner, but is not changing, so is preserving the expression of the protective ones. And we observed the similar results in both human islets and in stem cell-derived islets. Next. So we focus the continuation of this work to check the PDL1 expression on beta cell surface. So the programmed cell death ligand 1 is a protein that is expressed by beta cells, and this protein can interact with its receptor, the programmed cell death 1, PD1, that is expressed in T cell, mainly CD8 T cells. And the binding of PDL1 with the PD1 leads to T cell suppression and inhibition and a decrease in cytokine production. It is known that PDL1 is upregulated in type 1 diabetes individuals with remaining beta cells, potentially providing some protection against immune-mediated cell destruction. Next. So we want to check the PDL expression by flow cytometry using the INDEC beta H1 cell line. And we observe in the green bars that FGF inhibitor induce PDL1 expression on the beta cell surface when the cells were treated with the inhibitor alone or even in combination with interferon gamma. And we can see in the lower panel that the PDL1 expression on the beta cell surface occurs in a dose-dependent manner. Next. So, given the importance of PDL1, we were wondering whether this effect of the FGF inhibitor could protect beta cells from autoimmunity recognition by the CD8 positive T cells. And to answer that, we perform a co-culture experiment. So we use stem cell-derived islets that express HLA-A2. These cells were pre-treated with interferon gamma or the FGF inhibitor plus interferon gamma for 24 hours. And we co-culture together with pre-proinsulin specific CD8 T cells that are able to recognize the HLA-2 expressed by the beta. And after 4 hours, we check by flow cytometry T cell activation that was observed by the positivity for CD8 and CD107. So we can see in this graph, the gray bar represents the interferon gamma treatment that leads to an increase of around 40% in the T cell activation. And the FGF inhibitor plus interferon gamma in green is decreasing by 25% the interferon gamma effect. So we conclude that the inhibition of FGFR1 in stem cell-derived islets decreased T cell activation in this model of co-culture experiment. Next. So, as conclusions in our paper, we were able to integrate transcriptomic data from the target tissues of autoimmune diseases affecting beta cells, so type 1 diabetes, thyroid with Hashimoto's thyroiditis, brain in multiple sclerosis, and the joints in rheumatoid arthritis. We identified the commonly upregulated pathways that were associated with interferon signaling and antigen presentation. Then we confirmed the role of interferons in thyroid tissues using RNA-seq data of human cells, human stem cell-derived thyroid follicles that were previously exposed to interferon alpha or interferon gamma. And we performed the analysis of commonly upregulated inflammatory gene signatures, and we identified the FGFR inhibitor as a potential candidate to counteract the core inflammatory transcriptional program. And in the final part of our paper, we validate the FGFR inhibitor, the drug called PD703074, as a promising drug for preserving expression of beta cells, protective genes, and reducing their recognition by PPI-specific CD8 T cells. Next. And we would like to thank you all for your attention and also the Sugar Science for the opportunity, as well as our collaborators, our supervisor, Professor A. Zirik, and the financial support that help us to develop this work. Thank you. We'll be happy to answer your questions.
Thank you so much. Can you just stop the slides for a minute so we can see you guys?
Okay.
That's great. So both of you. Yeah, wonderful, wonderful presentation. Great finding that you have found a drug that's already available that could potentially be repurposed in this way. I wanted to open it up to questions. If anyone wants to drop a question into the chat or just raise their hand, unmute themselves.
I have a question. I have
Yeah.
many questions actually. So
Great.
I'm Geeta Parthasarathi and I work at the Tulane Primate Center. So I work on Lyme disease, and I have shown that FGFRs and FGFRs mediated neuroinflammation is important for Lyme disease in neuroborreliosis. But in Lyme disease, we also see interferon alpha is the signature that persists in patients that have persistent symptoms. And so this kind of makes sense to me that, both FGFR and interferon alpha would have a commonality there. So then my questions were like in the human samples that you all have, what stage of the disease are they all at? Because multiple sclerosis has many, many stages. So I'm not clear on which stage of multiple sclerosis were these patients even the samples were at from. That's my first question.
Yes, for the multiple sclerosis, from the original paper, they divided into early onset and late stage. So I was focusing on the early stage.
Okay, and the same thing for all the different diseases. Are they all from a particular stage of the disease, or is that something that's hard to get?
So for the type 1 diabetes, for the FACS sorted one, from the original paper, we have 4 type 1 diabetes donors, and as I remember, 3 of them under 3 years duration, and only one like 7 years. So put together, and
but they all had the same signatures.
Sorry.
But they all had the same signatures.
So we analyze it together. Generally, our results based on the fold change between the disease versus control. So we analyzed it together since we have limited number of donor, which is very rare and very precious. So, yeah.
Yeah, I know. I work with human samples and it's really hard.
Yeah.
So the other questions that I have was, I am familiar with PD, but PD is not just an FGFR1 inhibitor. It can also affect VEGF. So is that something that you have thought about and, discarded or disregarded? Is that something that you have checked to make sure that some of the effects are not through NFGF, VEGF mediated effects?
Yes, so basically in this concentration that we are testing, it works, is more selective for the FGFR1, but we are also comparing with using siRNAs for FGFR1. So then we can knock down this pathway and we observe the similar results. So showing that this effect is through the FGF inhibition.
Okay, thank you. And the other question, sorry for taking too much time. The one other question that I had was when I was looking at your how FGFR inhibitor, I don't know if you can show me put up the slide. The FGFR1 effects on interferon alpha and this one. So what I'm a little confused by is, so you have one that's what are the two different things? So one is interferon alpha at 0, 1, 5, and 10. And what is the other one? What are the...
No, sorry. So the white bars that are the zero is so the first white bar is a non-treated condition, and then the 1, 5, and 10 are the different concentrations of the
No, I got
inhibitor.
that. But I'm saying the two white bars, why are they? So let's assume it's just take any one of the graphs, HLA, ABC, or whatever the first one is, mRNA with the interferon alpha. You have a white bar that's not treated, that's 0 in both. So one that has interferon alpha, then there's the one right next to it. So why are the two white bars so significantly different if they are both 0? That was my question.
Okay, it's because they are both zero of with the FGF inhibitor, but the second bar, the one that is having the induction, is with interferon alpha, without so with zero of
Oh, I
PD.
see. Okay, now I got it.
Yes.
Yeah.
And
Okay.
then the 1, 5, and 10 is with the inhibitor plus interferon.
Oh, okay. Now it makes sense. Okay. Thank you so much. All right, I'm done.
Thank you.
Great, great discussion. Okay, so just to kind of like encapsulate this, right? You guys have the FGFR inhibition that results in a decrease of HLA with a canonical ABC. So you kind of like got an MHC1 invisibility cloak going. Then you get an increase in HLA-E, which is like the NGK to a break, right? So you break that thing, stop that. And you have so I want to ask you, do you think there is like then you have a reduced CTL and NK engagement. But do you think in between the break and the reduced engagement, do you think you have like any kind of potential support from HLA-G, even like HLA-G, the leader peptides, if you know what I'm talking about? The HLA-G and HLA-E work together. There's been papers in the past, Vincenzo Cirelli and Laura Crissa had paper regarding the HLA-G and its localization in the beta cells, and I think also alpha cells and the ductal cells. And so, how might HLA-G be playing a role in here with the HLA-E changes?
So we didn't check HLA-G expression yet, but it would be great to check these non-classical HLAs that are protective as the HLA-G and as well the HLA-F
Yeah.
to see if they play a role in the FGFR1 signaling. And here we observe the increase of HLA-E expression, but it would be nice as well to see the at the cell surface. So I think we have a lot to continue studying with this treatment and the delays.
Yeah, and I think sort of off camera, you kind of referred to some work you guys had done where you saw in human islet tissue, you stained for, HLA-G, and you saw it associated with cells that were still standing after the autoimmune attacks. That is very cool. Very curious,
Yes,
right?
yes, yes.
So yeah, that's really interesting. I wondered also like about the NK cells, like beyond your T cell assays, did you ever check to see whether or not you had any like NK, degranul like what's their phenotype after this FGFR1 inhibition? Can you see it or not yet?
Not yet, because we're focusing in the effect of FGF inhibition in the beta cells, because we are treating the beta cells.
Yeah.
So we didn't check other immune interactions yet, but we check the release of MIP1 beta from the CD8 T cells, and we saw a slightly decrease. So it would be nice to investigate in the future also the effect of FGF inhibitor in immune cells, since this receptor is expressed by the immune cells as well. So I think would be very important to
Yeah, sort of the
check.
downstream. Um, And then I was just wanted to ask you guys some questions about like, so this is very interesting to maybe repurpose this drug for those who are still in the prodrome and not yet diagnosed with type 1 diabetes. How might you see this used in the islet implantation space?
And so we check the expression, for example, of PDL1 in our beta cells and for 24 to 72 hours. And when we have the drug, so the treatment is ongoing, we can see the PDL1 expression. But after the moment we remove the drug, after 24 hours, the PDL1 expression starts to decrease. So, if the main effect of FGF inhibitor in protecting beta cells is through this PDL1 at the cell surface, so maybe will not be the best approach for cell transplantation.
Mm.
But I we are still investigating the mechanisms and how it works. So I think there is a potential for transplantation as well.
Yeah, it seems like you might be pre-prime a site, before you add the islets in or something along those lines. I'll leave that to you guys, the experts, to figure it out. But I guess my last question is, when we talk about like assisting, people who have one or two autoantibodies to, keep them sort of in remission prior to their diagnosis of type 1 diabetes. What kind of windows, have you thought about this? What kind of windows might be the best window to use this repurpose? I mean, this is really going far down the line, really just sort of like thought experiment. Use this, FGFR inhibitor to kind of keep things quiet by itself or with even another drug.
Yeah, I think the best timing it would be in the beginning of the disease, when we still have the beta cells to be protect. And as we discussed in the paper, it would be nice to test the FGF inhibitors together with interferon signaling blockers, such as TYK2 or JAK1/2 inhibitors, because it seems that the FGF inhibitor is not working the same way as the interferon blockers. So I think it would be nice to test both drugs together in the beginning of the disease and see how it goes.
So if they are already approved, right, those two drugs, maybe if you had a cohort of like stage 1 individuals and you could get them to, how quickly could you put that together?
Can you repeat? Sorry.
Suppose you were able to identify a cohort, a group of, stage 1 patients. And then if you already have these two drugs that are already approved in other settings, like how quickly could you get that clinical trial going? Would it be, or could doctors I don't know what the European situation is, but could you try to do off-label situations for physicians?
Okay, I think for the FGF inhibitor, we still need to work a lot because it's used mainly in cancer, and the safety and the toxicity is well investigated in adults and not in children. So I think
So you need some stage 1 adult.
yes.
What do you think, Desia?
No, I agree with Priscilla. And I think this question should be asked to Lilly and to the other companies, because in general, they're very reluctant to combine drugs, and the TYK2 and JAK inhibitors and the FGFR1 inhibitor are from different companies. But as Priscilla say, I think the JAK and the TYK2 inhibitors are much more advanced in the pathway to clinical use. I would first start with these drugs and consider FGFR1 inhibitors at a later stage.
Yeah, an add-on maybe.
Can I chime in? Because I think the doses are going to be far less than what you would use for cancers. You would need either, I don't know, they need to make a formulation because these are all orally bioavailable drugs. I mean, I'm in this field, so I can tell you this. So because these are all orally bioavailable, but the doses that they formulate for cancers is a lot higher. You might have to have them reformulate the drugs at a lower concentrations, but that is something that you got to talk to the company about and then see if it's something that they can collaborate with you to provide you the drug at a lower dose. Because this is something that I'm going to do for my own disease, because I also work on FGFR inhibitors. So I do have another question if nobody else has one.
Yeah, go
Don't
for
wanna
it. And
take
then
up
we
too
have
much.
one more in the chat, so sure, of course.
So I was kind of curious because this is for Xiaoyan. So in the beginning, you said that there's a lot of transcriptomic signatures that are similar between the three diseases or whatever, right? So I was curious, so do you know if the patients with multiple sclerosis, for instance, do they develop type 1 diabetes later in life? Because I don't know if these are autoimmune diseases occur in isolation, or is that something that they are connected? So is there a possibility that if somebody had multiple sclerosis, they will develop type 1 diabetes down the line because the signatures are similar? So I was curious about. This is more of an epidemiology question.
Yes, I think so, because as you may say in the introduction, many studies have proved that these autoimmune diseases they also share a significant overlap of risk genes. Many of them are related to HLA class 2. So I think it may have co-occurrence between these autoimmune diseases.
So in your cohort then, do all your separate entities, that's multiple sclerosis and your Hashimoto disease and the type 1, do you see an overlap in symptoms? Do you see an overlap there? Or these are separate concrete cohorts?
I think they are separate studies and there is no overlap. We just collected the data from different studies.
I just wondered if they if that was a factor that they eliminated when they picked the patients.
I think so. This is mentioned in the original paper.
Okay.
Just, I think your question is very relevant. As Xiaoyan say, many of these diseases have candidate genes up to 50% in common. And one of the mysteries in the field is why in some individuals it ends up with type 1, in others of rheumatoid arthritis, in others Hashimoto thyroiditis, and so on. But the autoimmune diseases most frequently co-occurring with type 1 are celiac disease in around 10% of cases, Hashimoto thyroiditis between 2 and 5%, if I'm not wrong, and the others are rarer, but are still more frequent than in the general population.
Yeah, that's why I was wondering, because I was wondering if the populations that were looked at as genetic signatures and transcriptomic signatures, are these like discrete, independent disease patients? So these are, for instance, these are Hashimoto patients, but they don't have type 1 diabetes yet. These are multiple sclerosis patients, these don't have type 1 diabetes yet. Or is there like an overlap, and that's why you're seeing something very similar between all three of them? That was my question.
No, there is no, in this disease, in these papers, there are no mention of overlap.
Okay.
I think they were isolated.
Interesting. Here's a question from the chat from City of Hope, Brian Liu. Thank you for the great talk and congrats on the GI paper. 2 questions. Are your SIG and HLA findings general to inflammatory microenvironments? Specifically, do alpha cells or exocrine tissues show similar ISG/HLA responses? Do you guys want to answer that one first?
Yes, I think this question might be our very interested next topic, comparing alpha and beta. And for this moment, we have the result that from the basal condition, alpha cells show a significant higher IST score compared to basal beta cells. And regarding the responses, I think because now we are expanding our data sets, and in the coming days, we will have the answer. Yeah.
Nice, exciting times. Good. And number 2 question from City of Hope. In your in vitro setup, only the FGFR inhibitor was added. So do FGF family members also increase the PD-L1 expression? I think you guys answered that and said it was specific, but.
Yes, it seems to be specific, and we are now working to check the mechanisms that are involved in the FGF receptor inhibition.
Well, it's exciting times. Just a great talk by the both of you. Just as again, following up on the earlier talk we hosted with Eugenia. And so it appears that the Zurich lab is really full steam straight ahead, just doing a lot of wonderful work. Hats off to everyone there. And thanks again for joining us and sharing this exciting new work.
Thank you very much for the opportunity.
Thank you. Have a great rest of your day.
You too. Bye.